So, good afternoon, everyone. I'd like to start my presentation by saying thank you to everyone here for making the time to participate in this very important endeavor. Let's see. I'd like to give special thanks to the National Academies and the committee members for inviting me to speak today and spearheading this effort. And to thank the TMD patients who've traveled here today, some with much difficulty and discomfort, to participate and share their experiences, challenges, and vision for the future of TMD clinical treatment. And lastly, but certainly not least, many thanks to the TMJ Association and Terry Cowley, who's been an unyielding voice for the patient community. So for those that are not familiar with NIDCR, our overall objective is to advance the nation's oral health through research and innovation. We support a broad research portfolio that spans the basic translational and clinical research arenas, as well as the science of implementing scientific findings into communities. We also support research training and career development programs to prepare the next generation of researchers. In addition to our outward-facing roles in the scientific community, NIDCR plays a leadership role in many collaborative efforts with other NIH institutes and centers, as well as collaborative efforts that span many other federal agencies. I've listed 5 of these efforts on this slide that you should know about, but in the interest of time, I'm not going to delve deeply into any of these except for the last one highlighted in yellow, which is our recently stood up Helping to End Addiction Long-Term, or HEAL initiative, that is targeted to combat the dual crises of opioid use disorder and pain plaguing our nation. The HEAL initiative has a congressional allocation of $500 million a year, with roughly half of that targeted toward research to enhance pain management. I'd encourage all of you to visit the websites and learn more about these efforts. Oops. So I've told you a bit about NIDCR's mission and how we fit into collaborative efforts across NIH and other agencies. But what does that mean for NIH's overall pain research effort? We have a long-standing commitment to funding pain research, and the bar graph on the left shows rising funding allocations to pain research through the years. In 2017, the NIH invested $516 million into pain research, with $440 million of that covering chronic pain research topics. This represented about 1.5% of the NIH's total budget that year. So while data for 2018 hasn't been released yet, I can tell you that the upward trend has continued and is set to jump quite dramatically in 2019 because of the NIH HEAL initiative. If we look at the distribution for pain research funding across all NIH institutes and centers, you can see that NIDCR's slice of the pie is about 4% of the total. If we look at how the NIDCR projects fall out by type of research being conducted and funding mechanism, you can see that 53% is basic science focused, 10% translational research, and 37% is clinical research, with the majority of these research project grants being research project grants such as R01s, R21s, and U01s. Let's see. If we look at how these NIDCR pain research projects parse out by disease condition, we find that NIDCR supports research relevant to a heterogeneous group of orofacial pain conditions that are complex, with many having unknown etiologies. And you can see here TMD has the largest slice of the pie. Research across these conditions is diverse and includes, but isn't limited to, investigation into aspects of biopsychosocial and genetic determinants of pain, neuroimmune interactions, sex differences, model development, chronic overlapping pain conditions, behavioral science approaches, and transition from acute to chronic pain. So if you look at this pie chart looks really similar to the last one. It's because it's the same thing, only with the TMD slice grayed out. I did that because I'd like you to think more broadly about NIDCR's pain portfolio, and specifically about how the pathophysiological mechanisms underpinning each of these disease conditions may be interconnected, and how research findings in any of these could have a significant impact on advancing TMD science. And I give you a few examples of some of these projects that fall into the neuropathic and broad categories shown there, such as defining generators of spontaneous trigeminal pain, developing animal model for stress-induced transition from acute to chronic pain, and sex differences in serotonergic modulation of trigeminal pain, all of which could have a bearing on TMD research. Okay, so keep that in mind as I show you NIH's investment in TMD research, which is captured as a standalone category for TMD research and captures both TMD pain research and non-pain focused research. We have a long-standing commitment to TMD research. The bar graph on the left shows funding allocations from 2011 through 2017. In 2017, NIH invested $13 million into TMD research in this category and funded 40 projects. So again, I can't show you data for 2018 because it hasn't been released yet, but I can tell you it denotes the beginning of an upward trend that we also expect to be bolstered by the HEAL initiative and NIDCR-led activities. If you look at the distribution across the NIH institutes and centers, you can see that NIDCR has the largest slice of the pie at 87% of this. If you look at how the projects in the TMD category fall out, again, by research type and funding mechanism type, you see 57% are investigating basic research, 6% translational research, 37% clinical research, again, with the majority of these being research project grants, such as R01s, R21s, and U01s. So what types of TMD research does NIDCR fund in addition to those considered to be pain-focused? We invest in a wide breadth of areas with the goal of improving the health of those who suffer from TMDs, including genomics and proteomics, systems biology to define the role of nervous, immune, and circulatory systems in jaw function, tools and technology, joint mechanobiology, regenerative medicine, behavioral research, and dissemination and implementation research. So just to give you an example of some of the basic research funded through NIDCR, I picked a couple of relevant publications. This by no means captures the breadth of all of our basic research relevant to TMD, but it shows you a nice cross-section of our broad research activities that impact directly on advancing the science of TMD. For example, one of our trainees was lead author on a publication investigating the neurocircuitry of orofacial pain to better understand why patients often report facial pain as more severe and emotionally draining than pain in other parts of the body. They found a previously undiscovered pain pathway that conveys facial pain signals directly to a key emotion-related node in the brainstem. Another NIDCR-funded investigator was part of the group that discovered that different immune cells mediate mechanical pain, hypersensitivity, in male and female mice, providing insight into mechanisms responsible for sex differences we see in many chronic pain conditions. We also see sex differences in the mandibular condylar fibrocartilage chondrogenesis, as denoted by the publication on the top right, indicating that estrogen promotes chondrogenesis and inhibits degeneration in female mice. And finally, Dr. Ha Yao's group, who is present here today, is pushing the field forward by investigating three-dimensional human TMJ muscle attachment morphometry and examining its impact on TMJ mechanics. I'd also like to highlight a recent publication from NIDCR-supported investigators that have developed a tissue clearing technique whereby bone, including the mandible and teeth, can be turned transparent with intact internal structures, allowing for 3D visualization of dental implant tissue interface, an investigation into the likely success or failure of implant integration based on successful blood vessel development and bone formation around the implant. In this study, the investigators found that while blood vessel formation occurred at comparable levels around both titanium and steel implants, successful bone formation only occurred surrounding titanium, indicating that the latter would be a preferable implant material. This vein of research is quite exciting and a valuable new tool for studying tissue-material interactions. Another area of NIDCR-supported research that holds therapeutic promise for TMD patients is tissue engineering focused on temporomandibular joint disc regeneration. While there's still quite a ways to go in this field, NIDCR-supported investigators are making exciting advances in this field by creating scaffold-free tissue-engineered implants that are similar to the native disc. Their work is currently focused on determining efficacy in repairing disc thinning in mini pigs, but one can see how this type of research is paving the way for development of tissue-engineered implants as clinical treatments for TMJ disc thinning and perforation, treatments for which are currently palliative and not reparative. I was going to talk a little bit about OPPERA, but I think we've heard quite a bit today, so in the interest of time, I'll skate through this slide, some selected publications, and on to the next. So what I want to show you in this slide are examples of NIDCR-supported TMD clinical research that include a study on the effect of genetic polymorphisms on responses to propranolol therapy in TMD, also known as the Soprano study, a preliminary safety study of botulinum toxin for treatment of myofascial TMD pain, and a study on the genetic effects of expectancy-induced analgesia, also known as placebo, in TMD patients. Some non-pharmacologic studies that NIDCR supports are a study determining whether high-definition transcranial direct stimulation can offer lasting pain relief in chronic TMD, and a patient-centered web-based program to educate and engage TMD patients in a personalized program, which you also heard about earlier today. So where does that leave us? What are the lessons learned, and where are there some opportunities to move forward in? So studies indicate TMDs are multifactorial conditions influenced by genetics, sex, gender, environment, and psychological factors. But significant gaps remain in our mechanistic understanding of peripheral and central mechanisms, adaptations, and psychosocial influences that promote chronification or resolution of TMDs. TMD patients with the most severe symptoms exhibit the greatest burden of comorbid conditions, pointing to a multisystem disorder. But the absence of a firm mechanistic understanding of TMD and a clear etiological targets has precluded development and clinical implementation of safe and effective evidence-based treatments. TMDs are highly complex, necessitating patient stratification approaches to facilitate precision medicine approaches. TMD models that recapitulate human pathophysiology are critical to development of safe and effective therapeutic interventions. And determination of the mechanisms that sustain or promote resolution of chronic TMD pain and strategies to intrinsically and/or extrinsically modulate these mechanisms represent therapeutic opportunities, as well as a fuller understanding of sex-based differences, influences, underlying TMDs will facilitate development of therapeutic interventions. And leveraging recent technological advances and resources developed through the NIH Brain Blueprint and Spark initiatives are poised to enable significant advances. With that, I'll say thank you and open it up to questions.